Hello, my name is Elena Matveishina. I work at the Institute of Bioengineering, and I would like to present my research titled Practical Guidance for Genome-Wide RNA DNA Triple Helix Prediction. Triplexes are triple helixes formed by RNA and DNA duplex, with Hoxtein or reverse Hoxtein interaction. Triplexes can be used by long non-coding RNAs to regulate gene expression. So it's interesting to predict triplexes. But this is a challenging task with great dependence on triplexes parameters. And two of existing tools for triplexes prediction, long target and triplexator, that is the best one, do not show great quality of prediction. So we want to improve triplex prediction quality, and to do so, we determine best triplexes parameters that should be used for triplexator. And also, as RNA tend to form complicated structures, but particular nucleotides can form Hoxtein bonds or Watson-Crick bonds, but not both at the same time, we speculate that triplex-forming nucleotides should be unpaired. So we would like to implement RNA secondary structure information to triplexes prediction. In this study, we used 4 lncRNAs, MEC3, TERC, HTER, and DACR1, and their DNA binding regions, determined by CHOP or ChIP-seq experiments. For each lncRNA, we predict triplexes using triplexator with different triplexes parameters, such as minimum triplex length, maximum error rate, and minimum G content of triplex. Then we measured triplex prediction quality with area under the ROC curve and select best triplexes parameters by the area. Also, we predict lncRNA second structure using two tools, RNA pale fold and RXS, with different thresholds for nucleotide pairing probability. And then we predict triplexes with triplexator only for single-stranded fragment of these lncRNAs. After that, according to measure of triplex prediction quality area under the ROC curve, we select best program for secondary structure prediction and best thresholds for nucleotide pairing probability. Triplexes parameters that lead to best results in terms of area under the ROC curve are 10 nucleotide as triplexes minimum length and 20% as triplex maximum error rate. But according to G content, triplexes can be divided into two groups. Triplexes with low minimum G content, at least 30%, and triplexes with high G content, at least 70%. We see no reason for preference to any of values for minimum G content, so we suggest to try both variants of this parameter. On the next step, we predict RNA secondary structure with RNA pale fold and RXS tools, with different thresholds for nucleotide pairing probability. After that, for obtained single-stranded fragment of each lncRNA, we predicted triplexes with two best sets of triplexes parameters determined above. And we found that RNA pale fold outperform RXS, and best thresholds for nucleotide pairing probability are 0.5, 0.95, and 0.97. Here are ROC curves for best triplex prediction for entire lncRNAs and their single-stranded fragments. Secondary structure usage leads to the best prediction quality improvement in MEC3 case. Second best improvement results are for DACR1 lncRNA, while improvement for TERC and HTER lncRNAs is very moderate. To understand the difference in prediction quality improvement, we use triplex domain finder to find DNA binding domains, DBDs, regions in length RNA that actually form triplexes. So we found that for MEX3 there are lots of DBDs, and many of them have nucleotides with rather high probability of pairing. So usage of strict thresholds for pairing probability leads to a few DNA binding domains left. But they usually form triplexes with rather high number of DNA peaks, triplex target sites, TTS. In case of entire length RNA DACR1, only 2 DNA binding domains were found, and only one was left when single-stranded fragments of this length RNA were considered. While for TERC and HOTER length RNA, several DNA binding domains were found, and practically all of them were kept intact after triplexed predictions only for single-stranded fragments of these length RNAs. So we speculate that the reason for the improvement in performance for MEX3 and DACR1 length RNAs is filtration of false positive DNA binding domains. So to draw conclusions, we suggest to predict triplexes in this way. You should use triplexator with 10 nucleotides as triplex's minimum length, 20% as triplex's maximum error rate, and 40 or 70% as minimum G content. You also should consider not only entire length RNA but also single-stranded fragment of length RNA predicted by RNA pair fold with 2 thresholds for pairing probability, 0.5 and 0.95.